Next topic is principle of flow cytometry. Like a microscope, which produce an image, a flow cytometer that gives multi-analysis by automated quantification according to set parameters. Exactly like microscope, with microscope we are able to see the cell in detailed structure. Here we are able to analyze the cells in set characteristics in multiple ways. It is able to analyze several thousand cells in a small time having specified properties. And when suspension is run, sheath fluid is used to pass one cell at a time through a small nozzle. So cell passes through this instrument one by one in a single file. And fluorescent dyes, they are used to identify different cellular components. When these label cells, they pass through the nozzle, the fluorescent molecules, they get excited, and when they get excited, they emit light when they come back to their normal or resting states. These emitted light signals, they are detected and then they are digitized for computer analysis. Use of multiple fluorochromes, they allow several cell properties to be measured simultaneously. So एक जो मॉलिक्यूल होता है उसको हम एक कलर में स्टेन करके किसी दूसरे मॉलिक्यूल को दूसरे कलर में स्टेन करके और इसी तरह किसी तीसरे मॉलिक्यूल को तीसरे कलर से स्टेन करके हम एक साथ सेल में कई मॉलिक्यूल्स को या कई जो कैरेक्टरिस्टिक्स होती हैं उनको स्टडी कर सकते हैं. And acquisition is the process of collecting data from samples using flow cytometer and attached software. This is the principle actually which is explained with the help of this figure. You can see that on the top, sample is loaded, which is consisted of different types of cells. Here they are shown in blue and red color, but as they pass through this instrument, they pass in a single file. You can see at the bottom, blue cell is coming down, then on the blue cells is the red cell, and so on. And on the left side, laser light source is shown, and this laser light that excite the fluorophore which are used for staining of these cells. And on the right side, there are different detecting mirrors and other instruments with the help of which we are able to measure that light which are emitted from these molecules. So when cells enter in this instrument, they pass through single file, and this sheath fluid that helps in passing these cells in a single file. Antibody staining can be used. What is direct antibody staining? If we stain the cells with directly labeled antibody, but we can also use indirect staining as well. In this staining, first cells are stained with unlabeled primary antibody, and then the cells are stained with labeled secondary antibody. And in this way, the cells, they are stained in two steps. I mean, first step with primary antibody, which is unlabeled, and then with secondary antibody, which is labeled. So both staining techniques can be used. Common fluorophores which are used for staining, they include propidium iodide, fluorescein, phycoerythrin, and many others. The ability of an antibody to resolve a positive signal depends on fluorophore which we use for staining the cells. And in this way, the cells are analyzed on the basis of different characteristics.